Hi everyone, Chris here. Hi. Uh, Today we'll be doing the virtual lab for exercise 22 in your lab manual, looking at population growth, and specifically we'll be looking at population growth in E. coli. And we wanted to film this to kind of, show you the techniques and, give you a visual of everything going on. And then this way too, you can get data and make conclusions and, analyze something to do with this lab. Like I said, exercise 22 is about population growth, and so population growth, as most of you are probably aware, populations have something called a carrying capacity, and that could be due to multiple things. It's going to be covered more heavily in lecture, and so resources, space, food, if there's something atmospheric, oxygen levels, something like that, pH in this case, we're going to be testing that. And then just, so we're doing a time one also, over time, given unlimited resources, you would think that things would continue to grow, but eventually there'll be a carrying capacity, whether that's space or whether the resources are actually limited or however. And so I'm not going to bog you down a whole lot with the calculations and everything. Suffice to say that when you get to the top of page 239, it wants you to kind of do a theoretical estimation of the E. coli, the population size. And so if you read the section before, it talks about how, the population can theoretically double in each instance. So what I want you to do for that is, just double those numbers for the theoretical and then compare them to the actual, which, is pretty interesting. And so those actual numbers are what we would typically see, if you graph them out, when something reaches carrying capacity. So, feel free to graph them out to give you a visual, because the numbers themselves don't really mean much until you kind of visualize them, right? So, that being said, I'm going to be going through procedure 22.1 and procedure 22.2, and I've already done a lot of the prep here. So 22.1 has to do, flip the page here. 22.1 has to do with measuring population growth over time. So if you were to make a graph, which you should be doing, you would put, so on your y-axis, you would put the actual absorbance value in this case is what we're going to be going with, and I'll explain that here in a second. And then on the x-axis, you would put time. So the absorbance value, so what we're doing here, and it says it in your book to record at 600 nanometers. So what that is that's a specific wavelength of light, right, that we are doing these measurements at, and 600 has just become the norm for, doing bacterial populations in broth. I'm not fully aware of why that is. I know it does have something to do with the fact that the different nutrients that are in nutrient broth aren't really picked up at 600 nanometers. So you don't have to worry about, if the broth is older or if it's more concentrated or things like that. And then you can use water as a blank, which is really nice. So anyway, we're going to, run the spectrometer. So what I've done here, don't worry about these ones yet, but what I've done here is I've already put my different times into cuvettes. So what I have here, I have 0 hours. So I don't know how clear that'll be. You can see that pretty well, I think. Doesn't focus very well, but that's fine. And then I have 4 hours, which again, it's so that I notice that the color is a little bit lighter, right? But the turbidity is what we're looking at here. So it's a little bit cloudier than 0 hours. And then I have my 8 hours, and again, you may not be able to see it that well, but it is a little cloudier, a little more turbid. 12 hours, and then 24 hours. And then what I'm going to do here is I'm going to show you how I filled these with the 48 hours, so that way you have an idea of how I'm going about this. So what I'm going to do is I'm going to grab my micropipetter here. So this is actually a larger one, so what I'm going to do is pipette out a mil, which is 1,000 microliters on this. Those metrics, right? And so what I'm going to do is I'm going to get a tip, which most of you have probably already done this in lab. This tip box kind of stinks, but that's fine. So got my tip. I'm already set at 1,000. I'm going to open up my 48-hour tube. And I'm going to slowly pull out. It's important that you do it slowly so it doesn't all splash back up at the pipetter, because then you have to sterilize everything. It's a pain because there's bacteria in here. And then I'm just going to dispense that mill into here. And then dispose my pipette tip properly. So the reason that we use these cuvettes, and I'm going to go ahead and cap it, sorry. And so the reason that we use these cuvettes, and you know it's important to use a very clean cuvette, and I'll just go ahead and show you the 48 here while I'm talking, and it should be a little cloudier, right? And so the reason that we use these is because they don't refract the light or reflect the light that the spec is going to be using. So it's pretty clear, right? So if we were to use something that was really cloudy or something that, you know, had a lot of, reflected the light, then that spec would obviously give us a really bad reading, right? The talking of the actual plastic. And I know that we've used in the past actual, I believe they're made of quartz cuvettes, which are very expensive. Just because they're more scratch resistant and give you a better, more accurate reading, right? Higher precision. So, all that being said, let's go ahead and start using the spec. So you can see the big machine that I have here set up to my left. What's really nice is we have a very nice Biomate Thermoscientific spec that actually has a really nice screen interface. So normally what you would do is you would run each one and kind of get your data, write it down somewhere, and then do your graph later. This one will actually give you the data as you go through it. So you can save a file. I actually have my flash drive plugged into this, so I could pull those files out and send them to you, which I plan on doing. And it, it's just a lot nicer, and it actually has a touchpad interface and all that stuff. It's very fancy. So how do we use this? So I'm gonna go ahead and try to pick you up here. Let's see how well this works. So you can see the interface here. I realize it might be a little bit out of focus, but basically what we're gonna do here is let me step back. So I've already selected what I was gonna do. So you could still run if you wanted to run fixed, or you could still run one at a time if you wanted to, but you don't have to. So what I've done here is I've picked OD600, which is, it's pre-made for running bacteria. It's gonna run at 600, but I can edit that too. And I'm gonna hit the plus, and then I'm gonna create a new session, right? So we'll go ahead and name this one real quick here. And let me set you down while I name it. So I'm gonna name this one 22.1 because that's what we're working on, right? So 22.1, and we'll go ahead and go Bio 102. And let's put the date on here, right? So it is March 30th when I'm recording this. So we'll go 3:30. I don't know where the hyphen is. There it is. Okay, so we'll go ahead and go with that. Done, hit continue, right? So we're starting with, like I said, 22.1. So with this one, I guess with both of them. So our blank is gonna be water. So I've already put water in a cuvette, and I realize that there's not a turbidity intensity for water on the thing, but that's fine. So what I ended up doing, actually, so my 0 hour, I gave it a turbidity reading of 1. Again, you've seen it. My 4 hour has a turbidity reading of 2. My 8 hour has a turbidity reading of 4. My 12 hour has a turbidity reading of 6.